Hi, my name is Zeynep Ulkan, and for my independent research project at Tallinn College, I explored the underlying mechanisms through which the antimicrobial peptide epinephrine-1 promotes migration and proliferation A375 cells, which were used as a human skin cell model with potential applications in wound healing. Wound healing is a vital biological process that restores skin integrity after injury, but chronic and impaired healing continues to affect millions of people worldwide and can also result in serious complications. Antibiotics remain the most common solution to treat suboptimal wound healing, but with antibiotic resistance on the rise, there's a growing interest in alternative therapeutics. One such alternative is antimicrobial peptides, which are small positively charged molecules that help the body fight infections and support tissue repair. These don't contribute to resistance the way traditional antibiotics can. One marine-derived antimicrobial peptide that has shown promising results is epinephrine-1. EP1 is a multifunctional peptide with promising anti-cancer and immunomodulatory effects, and recent work in our lab also found that EP1 significantly enhances migration in A375 cells, which were used as a skin cell model. Building on that research, I aim to further explore how EP1 promotes wound healing by investigating changes in cell proliferation, migration, and gene expression. Specifically, I looked at genes involved in inflammation and cell motility, which were NF-kappa B1, VIM, and MMP2, to better understand the underlying mechanisms of action. To begin, I identified the optimal dose of EP1 for the A375 cell line using an MTT assay, which showed significantly increased cell viability at 50 microgram per milliliter. This proliferative effect was further confirmed by a clonogenic assay, where the treatment group formed more colonies than the control. To expand on EP1's wound healing potential, a Boyden chamber assay was performed, revealing enhanced invasive behavior in treated cells compared to untreated controls. With the proliferative and migratory effects of EP1 confirmed, I next looked into relative gene expressions of the genes of interest through RT-qPCR. No significant differences were observed between the treatment and control groups for the expression of VIM and MMP2. This suggested that EP1-induced cell migration does not cause a shift towards epithelial mesenchymal transition, or EMT, as vimentin encoded by the VIM gene is an essential part of this process. The unchanged MMP2 levels further suggested that MMP2 is not a vital contributor to extracellular matrix degradation for migration, or that the mode of migration may be non-proteolytic. NF-kappa B1 expression was significantly increased in the treatment group. This upregulation suggested that EP1 promotes cell migration through activation of signaling cascades linked to NF-kappa B pathway. Therefore, it's possible that EP1 induces a pro-inflammatory response in A375 cells and enhanced cytokine production, triggering cell repair cycles and ultimately facilitating cell movement. The unchanged MMP2 levels were also confirmed through a gelatin zymogram, which was performed on serum-free media collected from the Boyden chamber assay. The zymogram revealed no significant changes in MMP2 activity, but revealed a significant decrease in MMP9 activity in the treatment group. This was unexpected, as the upregulation of NF-kappa B1 is associated with increased pro-inflammatory factors. However, it is possible that EP1 alters the cleaving processes and post-transcriptional modifications of MMP9 that allows it to be detected by the zymogram performed. Nonetheless, the concurrent upregulation of NF-kappa B1 and decreased activity of MMP9 in the treatment group suggests that EP1 induces an NF-kappa B-linked non-proteolytic mechanism of migration. To validate the findings of the study, further investigations should explore the effects of EP1 following gene silencing or gene knockout of NF-kappa B1. Overall, the findings of the study highlight the importance of further research and provide valuable insights into EP1's potential therapeutic applications.